Hi, everybody, and welcome to this week's lecture. What we're going to be focusing on today is going to be the introduction to viruses. So our learning objectives, or what we're going to try to get accomplished today, are going to be to describe the basic structures of our viruses. So exactly what they're made of, what they're doing, and what are some of those functions. We're going to look at and explain what term host range means and what that kind of entails and how they're going to recognize who they're going to infect. We're going to also describe a typical life cycle of a virus. And then finally, we're going to go ahead and check out some of the common viral infections that you most likely have come in contact with and kind of what's going on with them and what they do. All right, so let's get started. Viruses. Viruses are going to be really, really tiny particles that can infect. And one thing that's important is in order to reproduce, so in order to make more of these viruses, they have to infect a host. The reason why is because they're known as acellular and they are not going to be alive. So what they're going to essentially do is viruses, they're just going to be a bunch of protein shells with a capsid in the middle, which is going to protect the information of the virus. So what the viruses are going to do is they're going to essentially commandeer a host cell. Okay, so they're going to go ahead and they're going to find another living organism or a living cell, and they're going to essentially go ahead and infect it and reprogram it so it can become a virus factory. And what that basically means is it's going to go ahead and infect it, inject a lot of its information into this host cell, and then it's going to go ahead and utilize a lot of its resources in order to produce a lot more of those viruses. So that's how it's going to reproduce. Now, if you look down here, here are some examples of some viruses, and these are very, very famous viruses, especially in recent times. We have our coronavirus here, which is what it looks like underneath the microscope, and then we have more of a computer-generated rendition of what that's going to look like. Now, the SARS-CoV-2 or coronavirus is going to be 120 nanometers, so again, very, very tiny. Remember, viruses are going to be incredibly tiny pieces. And remember, they cannot reproduce on their own. They require a host cell. So viruses are going to infect something else in order to, again, reprogram the cell in order to produce more of the viruses. Because remember, viruses are not considered alive. And we'll learn more about this. You'll see that on the side over here, we have all of these kind of funny little circles, and those are going to be known as our spike proteins. And these are actually going to help the virus recognize what host cells they can actually infect. And we'll check that out in just a second. So now that we went ahead and looked at what a virus is, we're going to go ahead and take a look at what a virus structure is going to be. Now, looking at this over here, you can see that we have an example, which is going to be HIV, and we have a diagram here, and then we also have what it looks like underneath the microscope. Now, you can see we have some spike proteins right here and right here. And again, the spike proteins are going to be important because it's going to help the virus recognize who it can actually infect. We have the envelope right here and right here. And then it's just going to be the outer shell or the protective portion of the virus. We have the viral genome, and this is super important because this is all the instructions that the virus needs in order to produce more of itself. So you can see that the viral genome is going to be right about there. And the capsid, which is going to be a very, very important part of the virus as well, is because the capsid right outside here is going to protect the viral genome inside. So you can kind of think about this like a phone and then a phone case. Phones, really important. They can do a lot for us, hold a lot of information. Some phones can be fragile, so we typically put a case on it in order to protect that phone. Same thing. In this case, it's kind of like a phone case on the outside, and then the inside, the viral genome, is going to be the phone itself. So viruses, there are going to be thousands of different viruses in the world, many, many different types. And viruses, again, can vary in size, shape, and life cycle. Some of the shapes can be bigger or smaller. Sizes can be bigger or smaller. Shapes can be completely different. They're not always going to be round. They can sometimes be triangle shaped, things like that. Like if we take a biophage, they are going to look something like this. versus a circle. And then they are going to have different life cycles. Some of them only take a couple hours, some of them can take a couple days. Again, it kind of just depends on what virus we're talking about. So this is kind of what a breakdown of what the viral structure is going to look like. Again, despite the viruses and how they can vary in all their size, shapes, and things like that, they are going to have a lot of the same basic pieces in order to, again, recognize their host cell, so they can infect, and then how they're going to go ahead and produce more of them. So now we're going to go ahead and check out what a host range of viruses are going to be. So viruses can infect every type of cell. Now this is important because it's going to include essentially everything that's alive. Plants, animals, fungi, protists, bacteria, archaea, essentially anything that is able to be infected can be infected by a virus. Now, most viruses can only infect a cell of one or few species. So they're going to be kind of very specific. Again, they can only infect one or a few species. And this is going to be known as our host range. So again, host range, think about the range of hosts that it can infect. And most of the viruses are going to have a very narrow range and typically can only infect specific hosts. And the reason why is because of something known as our lock and key mechanism. So what's going to happen here, this is the example of SARS-CoV-2. So we have our virus right here. Again, it's not alive. It's not able to reproduce on its own, so it needs to find a host cell in order to reproduce itself. So it's going to look around and it's going to go ahead and find something that can fit this spike protein. You'll notice that there's this kind of irregular shape over here. But if you look closely, you can see that it can slot in to this host protein. So this is going to allow the virus, in order to connect with the host. So this is going to tell right here, this interaction is essentially telling the virus that, hey, this host right here is going to work for you. You can absolutely go ahead, infect it in order to reproduce and make more of yourself. So in this case, the spike protein of the SARS-CoV-2 is going to bind with the ACE2 proteins on the surface of the respiratory system or the respiratory cells in order to infect them. Again, they're going to be able to find the correct lock for the key in order to go ahead and infect them. So this is what that life cycle is going to look like. And we have about 5 different steps. So viruses, again, must take over a host cell in order to replicate or reproduce. So this is what a typical virus life cycle is going to look like. First things first is going to be that attachment. Remember, that spike protein is going to connect with the host protein in order for it to recognize it. So you can see attachment right here. We have that lock and key. So if that passes that check, then that virus is going to go ahead and connect to it, check it out, say, hey, this is going to work. So they're going to go ahead and affect it. They're going to go ahead and enter. So that means they're going to go ahead and start going into the actual host cell itself in order to start getting access to all of those resources to reproduce. So again, now we're going to have our replication. So the viral genome is going to go ahead and start copying itself. So what's going to happen is, remember, that capsid, that protective shell that's going to be protecting all of our DNA or all of our instructions, is going to go ahead and start to disappear. It's going to come apart. It's going to dissolve because now we don't want that, right? Once we're inside the cell, we don't want this capsid anymore because we want all of our genetic material to be free in order to go ahead and start replicating itself. So once that disappears, the gene is going to start copying itself. It's going to start making more viral proteins by using a viral RNA with its host ribosome in order to make, again, those proteins. And then once it has done that, it's going to go ahead and kind of come back together. And it's going to go ahead and take all of those copied genomes, and it's going to use all of the other viral proteins, again, all of the different structural components it's going to need in order to assemble itself. So now all of the new viral particles are going to assemble, and they're going to start to go ahead and get ready to exit the cell. And this is going to be known as release. So all of the new viral particles are going to exit and release. They're going to break out, and then they're going to go off and try to find new host cells to infect. So this is going to be a complete big cycle. So again, attachment, lock and key, it's going to go ahead and find the correct host cell that's going to fit the spike protein. It's going to go ahead and enter. It's going to go ahead and start replicating its genes right here. It's going to go ahead and start making more of its viral proteins, whatever it needs in order, again, to have that structure work. assembly, it's going to add all those parts together, it's going to leave, and then it's going to go ahead and start that cycle over again by finding a new host cell. All right, so with that being said, here are some common diseases caused by viruses. So some of these you most likely have had an interaction with, some of these you have probably heard of. One of them is going to be the common cold, rhinovirus. Rhinos are very known for having a very long tusk in the front. So rhino meaning nose, in this case, it's going to be the common cold. It's safe to assume that majority of people have had some sort of interaction with the common cold. Again, what it's going to do, it's a viral infection, and it is going to cause sore throats, ear infections, sinuses, things like that. It is highly contagious, and one of the big reasons why it's so contagious is, remember, that virus is looking for hosts in order to reproduce. Influenza. Influenza is going to be another one. There's two main types of these viruses, type A and type B. And again, it's going to be the seasonal, it can be combated with the seasonal flu vaccine. One of the cool things about vaccines is they're going to be essentially an attenuated, or in this case, a non-reactive, reactive version of the virus, so they are not going to replicate. So our body is able to recognize it when we do get attacked, and then it's going to be a way better way to protect ourselves. So we will have some of those antibodies in order to defend against those viruses. Chickenpox. Super contagious. Typically, you get an itchy rash, and it's going to have small fluid-filled blisters. They're typically going to be very mild, especially when you're younger, but once you get a lot older, they can be much more severe. Measles is going to be another type of virus caused by the rubella virus. Again, extremely contagious, and in this case, it's not going to appear until 10 to 14 days after exposure. Stomach flu, norovirus. The norovirus can cause vomiting and diarrhea, highly contagious, and you typically get this through contaminated food or water. And again, this is going to be typically or most frequent in closed and crowded environments such as hospitals, nursing homes, child centers, schools, and cruise ships. All right, and that is going to conclude our lecture. So there are going to be two-part review questions. What I'm going to do is I'm only going to go over some points that I really want to emphasize for these questions. So go ahead, I'm going to give everybody a couple seconds to pause this video, see if you can answer these, and then we'll go out and look over two of these specific ones, and then we'll do the same thing for the next slide. All right, welcome back. So the two that I kind of want to go over are going to be number 1 and number 2. So number 1, it says the blank is the protective protein shell surrounding the virus genetic material. That is going to be the capsid. Capsids are going to be very important because, again, its job is to protect that viral genome. This right here is all of the instructions it needs in order to make more of itself. So without this, the virus would not be able to reproduce. So again, this is super important. So the capsid is going to do a very, very important job, and it's going to provide that extra layer of protection in order to ensure that that genetic material is going to be safe and it's going to be carried to where it needs to be once it's ready to reproduce. Number 2 is going to be spike. So viruses are going to use that spike protein to attach to the surface of the host cells. Remember, the spike proteins are going to be really important. They're essentially going to tell whether or not it is able to go ahead and infect it or it is not. Because viruses are going to be so specific, the spikes are going to be very specific to those hosts. All right, so I'm going to give everybody a couple seconds again to pause this, and then we're going to go ahead and go over number 2. All right, welcome back. So what we're going to do is we're going to go ahead and take a look at number 2. So in this case, it says the snow virus is a new virus that has a spike protein shown below. So this one right here. Scientists know that the snow virus must bind to the rose protein in order to infect the cell. Using this information, scientists want to protect the host range of the snow virus. So in this case, it's saying using the information above about spike proteins and rose proteins to protect the host. range, which ones are going to infect which, essentially. So we know that this right here is going to be a spike protein. We have that triangle, and then we have this right here. So remember, these are going to be very specific to our hosts. So in this case, we need to see which ones it's going to actually be able to connect or fit into. In this case, the cat is not going to work because it's not going to be a snug fit. We go ahead and go to this one, the dog. It is a lot closer, but again, this is more of a U shape. So again, it is not going to work. In this case, if we did the pig, it would be correct because again, you can see we have this kind of triangle portion here and we have a triangle portion here. So that means the cow would not work. All right, and that is going to conclude our apply your knowledge version portion for these viruses. And that is going to conclude our lecture. So again, if you do have any questions, please feel free to ask, and I'll see you in the next one.